Thanks for the invitation to give this talk. In addition to my own experience, Kim, Mindy, and Phil each have extensive experience with experimental networks, and they've provided their own perspectives and input into this talk. So I've done my best to weave our thoughts and experiences together to address the topic at hand, which I guess I could describe as the special sauce that makes successful collaborative experimental networks. I'll start here. This is the scale of the Earth, where most of us at this meeting want to generate both understanding and prediction in a changing world. In this image, you can see the beautiful chromatic patterns that have been generated by stitching together data from decades of satellite missions. For me, this depiction of Earth it's both awe-inspiring and informative. These patterns tell us about the annual sunlight energy captured by ecological communities of plants across Earth's environments. And with satellite images carefully stitched together again across both space and seasons, we can visualize the seasonal shifts in Earth's primary production across both space and time. By adding in the seasonal patterns, we build from that static image on the last slide to visualize the importance of both precipitation and temperature for determining regional patterns of carbon uptake and loss by the vegetation. And while we could all, debate about whether and at what scale these retrospective data give us a decent first approximation of productivity on Earth, we all know that climate isn't the only current driver of regional productivity on Earth. Depending on who you ask, people at this meeting may be most concerned about the amount that we don't know about Earth's spatial and temporal responses to future changes in temperature and precipitation, or maybe the interactions of climate change with other ongoing global changes to, for example, nitrogen deposition, or to land disturbance, conversion, degradation, including conversion to pasture and rangeland, or even to the impact of changing biodiversity and composition of plants, of microbes, arthropods, even herbivores due to both species extinctions and invasions. My key point here, though, is that the Earth system isn't changing one factor at a time, and these many changes aren't proceeding in lockstep. So changes in a factor at one location may not co-vary with changes in another factor at the same location. And importantly, these factors may, in fact, likely interact with one another. So, for example, increased nitrogen could alter the nutritional value or defenses in the plant community. This may vary with the amount of precipitation arriving in that area. And this could change relationships with herbivores that may feed back to impact soil carbon. And these different factors may have different importance in different regions. So when we ask in this workshop, for example, how will climate and global change affect ecosystem carbon, water, and biogeochemical cycling and associated climate feedbacks? This is an exceedingly difficult type of question to answer because the answer is likely to be contingent on whether there are interactions between climate and other global change factors for our key response variables, right? At the strength, the strength and importance of the interactions may also vary by region. If we perform a factorial experiment to simulate future conditions of, let's say, altered precipitation, temperature, herbivory, or nutrient supply, or even a factorial combination of all of these, if we're insane and if we could, we're constrained to inference about how the vegetation and soils under a single set of conditions are going to be responding. So those experiments at single sites can give really important insights. They can inform models, but they can't tell us what will happen under different biotic or abiotic site conditions. So for example, this image in the bottom right is an image of my own field site. It is being performed under the conditions shown where the arrows are pointing in Minnesota. But what if I perform the exact same experiment with fences, with fertilization of different types of nutrients in Central Africa or in the Himalayas or in Central Australia? I might find radically different results because different factors are more important under different conditions. What we really need to strengthen our understanding of the links between Earth's biological responses to multiple ongoing changes that vary across space. And these Earth system models is a better understanding of those contingencies. Under what conditions do different factors matter most? One way to achieve this is by doing multi-site replicated experiments under a wide range of Earth's conditions. And because of the clear gap between single site experiments and scaling up to predict future responses across regions or continents, and because of the benefits of replicated work across those regions and continents, multi-site replicated experimental studies are being increasingly implemented in ecology. In fact, multi-site experiments that Kim, Mindy, Phil, and I have dreamed up or led or participated in include ITEX, the International Tundra Experiment, DroughtNet that manipulates precipitation, nutrient network that manipulates herbivores and fencing, and DragNet that I don't have an image of yet, that is experimentally manipulating nutrients and disturbance. By implementing these multi-site, multi-continent experiments, we're each working to fill the gap in understanding about the conditions that most strongly determine different responses. Responses like productivity, like soil chemistry, like diversity loss, or other key outcomes that we want to understand when and where and why they occur. There's an increasing number of distributed factorial experiments that are replicated across sites and conditions, and they're experimentally asking what happens at the intersection of multiple global change drivers. So what do the four of us suggest when designing a large-scale collaborative global change experiment? I begin, I guess, as we were all trained, with hypotheses. While it sounds kind of obvious, start with clear core hypotheses that are key to understanding and prediction of the focal responses, right? What's the greatest mystery? What's the most important relationship or critical response to document and track across Earth's conditions? Where is there a mismatch or lack of knowledge for the Earth system models? What's the time scale over which we expect it'll play out? Core hypotheses will help guide those future decisions as well. The clarity from them will help decisions about the spatial and temporal scale of the required manipulation to test the hypotheses, the minimum area or the response variables that are critical to collect, and the key regions that need to be represented in the data set. So always referring back to the core hypothesis or hypotheses will help keep future decisions, and there will be many, focused on achieving strong tests of those central hypotheses. The next piece of advice we have is to have simple, clear protocols. To have a replicated experiment that's directly comparable across all sites, it's really important to have identical manipulations at every site. Simple, clear protocols will help to achieve this. Try them out. Estimate expenses. Check on availability for every component on different continents. A simple design replicated identically across sites has other additional benefits too. So by designing a simple study that tests the core hypotheses, then allowing network contributors to build from this with new response variables or even treatments that don't compromise the original design, this can be a really powerful use of an experimental network. It's amazing how inventive people can be. And providing a simple platform that can be leveraged for this new work will generate a lot of great and unexpected science. Designing around clear hypotheses will help cost considerations too. Regardless of the experiment that's going to be performed, think it through really carefully. Keep it as simple as possible. The more difficult and the more expensive the manipulation and sampling, the fewer people will be able to contribute. Difficulty and expense will particularly reduce participation by scientists from underrepresented regions. So simplicity of design is a really important consideration for documenting responses in less well-studied regions. Consider the funding model for site participation. What will it look like in the Oak Ridge project? How will sites pay for their implementation costs? And what will those annual costs be? Again, this is a trade-off surface that's really important to consider. More sites or more intensity of the manipulations or sampling. We add that a consideration of costs for network scientists is really important because while there's clear recognition of the value of network science by the scientific community, funding agencies don't generally support or the creation or maintenance of networks. it's possible that the group at Oak Ridge can overcome this, but more generally, given the community-wide recognition of the value of this approach, funding for people and data management is an issue that we as a community need to tackle for network projects. Okay, while people may be excited to contribute to the experiment, not everyone has the same bandwidth for participation. In our experience, by providing opportunities for different levels of contribution, from a one-time observational data sampling, or maybe just a few core experimental treatments, or a few years of data collection, or even just a few core response variables, all the way to long-term replication of a factorial experiment with many different response variables, you'll help a wider range of people to engage. Again, the design decisions can be prioritized by aligning with the core hypotheses. What is the greatest value for the least effort? Regardless of the experimental design, this kind of work can't be done by a single investigator. It has to be done. This documenting of the same biological response variables across Earth's environments has to be done with massive collaboration. So always with an eye to the hypotheses, the more environments represented, the stronger the data set. And that means you're going to need to meet the needs of people that are participating in this study. And that leads to the second part of this talk. Scientists are busy people with both personal and professional needs. They are participating because they're curious, they're motivated, they're excited, they want to contribute to a larger understanding of the world than they can generate alone. The network benefits by drawing from the creativity of the contributors. But keep in mind that every network scientist already has a full life and can have a great career without contributing to your network. So start with one basic assumption in every decision you make, that everyone contributing to the network is busier than you are. That empathy is going to help guide decisions about experimental design, meeting planning, management of people, and management of data. Most of us here are trained in experimental design, but few of us have training in the soft skills of managing people to maximize science joy and productivity. So I'm going to focus on the key to success for a collaborative network, which is kind of the second one, which is building and maintaining trust through structure, decisions, and actions. And as the old adage goes, building trust takes time, but losing it can happen in an instant. This is true for network science too. So how do you build trust? By providing clear guidelines and expectations at every step, you don't waste the time of contributing scientists. That includes creating and sticking to clear, simple field protocols and expectations for laying out plots, data collection, data sharing, and authorship. Because of differences in science culture across countries, there can be some pressure to privilege certain network members over others. So keep in mind that everyone's working equally hard and has agreed to the core network rules and expectations. Bending or changing rules to privilege one or a few contributors may undermine trust by all. As in any relationship, clear communication is critical for building trust. That doesn't mean that communication needs to be constant, but rapid response times and clear answers when questions are asked is really, really important. Publications are critical for communicating discovery, and careers depend on spending time on projects that'll result in publication. However, with many creative people in a network, ideas may have some overlap. And here's where communication is critical to maximize new insights from the shared network data and collaboration among those that are interested in the same topics. That communication helps to minimize overlap and maximize new knowledge. Everyone in the network is spending their own valuable time and effort, and maybe even research funding, setting up and maintaining the experiment, sampling, and submitting data. While it might not be an obvious part of gaining and maintaining trust, effective data management and curation reduces errors, it minimizes wasted time, and it makes time spent on analyses efficient. For every investigator contributing to the network, seeing that the data they submit will be managed and curated well builds trust within the network. Everyone needs to feel that participation will help them achieve their professional goals too. Building social norms within the network by setting examples of inclusion through care and attention for the needs and goals of each individual. That's another way to build trust. So clarifying roles, encouraging contributions from everyone, and equalizing the playing field during meetings, those all set the tone for everyone. That extra sensitivity to those who may feel less comfortable. So, for example, early career scientists or those who don't speak English as a first language, that's also really important for ensuring that everyone feels welcomed and valued. Um, a happy group where every person feels valued and truly welcome, it really sets the groundwork for a productive group in which people are sharing and building from each other's ideas. The structure of the meeting also sets a tone. It can be designed to make participants feel really important and really valued by holding repeated meetings, getting everyone in the room talking about their scientific interests, I'd say early and often in every meeting. People feel more included. Breakout groups where individuals self-identify their interests and participants guide the direction of the work, that also helps to maximize productivity with the shared network data sets. But a really flexible agenda, but with daily check-in points, that also helps everyone feel that they're valued, that they can pursue their own creative interests and work with people with shared interests. They can meet their own professional needs. Additional awareness by the network leaders to make sure that everyone finds ways to engage. That shows everyone in the group, even those that are already engaging, that inclusion is a network value. That builds trust and it makes the group more productive. Okay, depending on the goals of your network, diversity is likely to be high if your network and efforts at inclusion succeed. You'll have participants representing, for example, many career stages, many different employers, skill sets, countries, languages, environments, scientific interests, and even lived experiences. So regardless of the employer, the degree of isolation, even the career stage, providing opportunities for mentoring and networking helps everyone to engage. This particular picture shows a group of grad students and postdocs from 3 different countries spanning 3 continents, all of whom were deeply interested in drought effects because of increasing droughts in their own field sites. None of them spoke English as a first language. They came together because of a shared academic and applied interest. But then they became a group that mentored each other and stayed in touch for years. So that leads to a related point that early career researchers are one of the keys to a network's vitality by setting a tone of support and of inclusion and providing opportunities for growth into leadership roles. A research network can be incredibly impactful for the careers of participants. It can serve to foster a next generation of scientists that are really exceptional collaborators and really understand how to effectively use the powerful tool of network science. Stepping back, large collaborations generate this unrivaled set of opportunities for new insights and discovery about, functioning of current and future biological world. Designing the large-scale collaborative experimental networks requires a sensitivity to what they can do really well and what they can't do, as well as other tools we have at hand to study Earth. The goal of the next steps for the new Oak Ridge experiment will be to dovetail with Earth system models. And knowing this, focusing on this, will help to focus and constrain the design of the work. It'll help to inform the spatial and temporal scale of treatments. It'll help to determine key response variables and methods for collecting those response variables. Here's some advice, which I guess was solicited when you asked us to give this talk. Inform the design by carefully considering the core hypotheses to be tested. But keep an eye on the balance of cost, of effort, and maximizing the number of sites or including sites in historically understudied regions. Build from the best practices that the group that worked on this talk, have learned in designing and leading distributed experimental networks. The experimental design is very important, and design for a successful network includes some special considerations that I talked about earlier. We've each had successes and failures. Don't reinvent the wheel where it's not necessary. Also, consider who you want to contribute to the new effort. Know every scientist and their professional needs and help them to achieve this through participation in the science of the network. Be careful to use soft skills to build and retain trust of those who choose to contribute to the effort. Have clear ground rules, for example. Make sure that you have an eye to inclusion at all steps. And it's important to never forget that every contributing scientist is a person. And the people who choose to be involved are the power of the network. It's not just the dots on the map. I'll end with the observation that for ecology, a field that frequently struggles with physics envy, this model of international egalitarian collaboration, it was once called the dream by the Director General of CERN, of all things. And I'd say that in the field of ecology, we're well on the way to figuring out how to achieve this, to make predictions about future Earth. And I'd agree with this statement. We do have a very powerful tool in our hands.